Hey Mustangs, in this video we're going to be taking a look at enzymes and their role in metabolism. So if you remember, metabolism involves chemical reactions, the breaking apart of molecules, rearranging of molecules. And if you break that down even further, chemical reactions, you can break them into two groups. You have catabolic reactions and anabolic reactions. Catabolic involve the breakdown of molecules, while anabolic involves the building of molecules. Enzymes are involved in your catabolic and anabolic pathways and reactions. So when we connect it to energy, catabolic reactions, remember, are exergonic. When you break apart molecules, you actually release energy. And then anabolic, when you look at an anabolic pathway, you are putting molecules together, you're building, and that takes energy. So that's endergonic. So you have exergonic and endergonic and catabolic, anabolic, so all that. You got to keep all that stuff straight. And now enzymes are going to come into play. So with all those chemical reactions, whether they're catabolic or anabolic, almost all of the chemical reactions that occur in your metabolic pathways involve enzymes. So with a chemical reaction, you have molecules that you start with. And these molecules are going to be the ones that are broken apart and rearranged into your products. Here you can see we have an anabolic. We're putting these two together, and these molecules come together to build this molecule here. So what you start with, the molecules you start with, remember, are your reactants. And the molecules that are built at the end are the products. Okay, so reactants are the molecules that are going to be broken apart and rearranged, and you have your products here. Okay, or it could be the molecules that are going to be put together. So in either case, really, these still get broken apart and rearranged into your final product. So you have reactants and products, and the arrow tells us that a chemical reaction is occurring. Now, chemical reactions are part of your metabolic pathways, so these are happening in your cells all the time. You have trillions of chemical reactions going on every single second inside of your cells. And these reactions are what make life possible. They're absolutely necessary. And if these stop, then you stop building the necessary molecules that you need to keep your cells going, to build your cells. So chemical reactions should never stop. They might temporarily be stopped by things like hormones, because remember, hormones change momentarily, temporarily, what a cell is doing. But for the most part, you never want these chemical reactions to completely stop. So the chemical reactions that occur inside your cells, many of them occur so slowly that life wouldn't even be possible. So there's a problem there. If so many chemical reactions, you need all these chemical reactions to keep your cells alive, how is it that they occur so slowly that life wouldn't even be possible? Well, it comes down to activation energy. So these chemical reactions, if you don't have enough energy, many of these chemical reactions won't even occur. So that's why we say they would occur so slowly. They need so much energy that they just would not happen fast enough to keep your cells alive. So the amount of energy needed in order to get a reaction started is called activation energy. So I'm going to draw a quick little graph down here. So if this is the amount of energy, and it increases as you go up, so more and more energy go that way, and this is the progress of the reaction. Okay. Let's say it's an exergonic reaction. So you're going to take the reactants, and it's going to be catabolic. You're going to break them apart, and you're going to release energy. So we'll say that the reactants start with this much energy here. In order to get those reactants to break apart and then release energy, those reactants, you have to put in some energy. So you put in some energy. Eventually you hit a point where there's enough energy for that reaction to occur. The reaction occurs and you get your products. So here is our reactants right here. And here's our products down here. And we know this is an exergonic reaction because the reactants have more energy than the products. And we know it's exergonic because clearly we've lost energy, and that's why the products have less energy. So exergonic energy has been lost. Okay. Now the amount of energy from here to here, that point where finally the chemical reaction started, that there is your activation energy. So that's the amount of energy needed to actually get that chemical reaction started, to actually break apart bonds and rearrange into what you're actually looking for. So activation energy is really, really important when it comes to chemical reactions. Some chemical reactions need more energy than others, so the activation energy can vary depending on which chemical reaction is going on inside of a cell. But in any case, even exergonic reactions like this, you still need to put in some energy to get the chemical reaction started. So exergonic, endergonic, doesn't matter. Energy is needed to get it going. All right, so that's where enzymes come in. Enzymes make it so that way you can speed up the chemical reaction. Exactly how it does that, we'll take a look in just a moment. So enzymes are a type of protein, so that's really important. Enzymes are a type of protein that speed up chemical reactions, and we call them a catalyst. So that's another word you can use for enzyme. So a catalyst is anything that's going to speed up a chemical reaction. Now there's two important points about enzyme that involve them and connect them with the activation energy. So first, enzymes only work with specific substrates. So looking at this word substrate, substrates, remember, are simply the reactants. So I could put in parentheses reactants. So all the enzymes that you have, they're very specific. So that's one really important thing about enzymes. Enzymes are very specific with what they can work with, what reactants they can actually bind with. And we call those reactants substrates. So substrates is just the fancy name for reactants when an enzyme is involved. The second thing, enzymes lower activation energy. So enzymes make it so that way a chemical reaction they don't need as much energy in order to get started. So it makes it, instead of needing this much energy for a chemical reaction to occur, with an enzyme you only need this much energy. So you need less energy for that same chemical reaction to occur. All right, so if we take a look here, this is the very important graph that you need to know and understand. So here's the amount of energy. The higher you go up, more energy. You go this way, that's the progress of the reaction. So I actually drew that already, and it looks really complicated. Activation energy with enzyme, without enzyme. So I'm going to simplify it. So I'm going to redraw it and simplify it here. So you have your graph. This is the amount of energy. I'm just going to put energy this time. So this is amount. I'll just put amount. So amount of energy, and this is progress of reaction. This is more energy as you go that way. This is going towards the completion of the reaction as you go that way. So I already drew this one. So you start with the reactants. You put in energy. Eventually there's enough energy to get the chemical reaction going, and it occurs. So you go from reactants to products. Once again, in this case, you can see it's exergonic because the products have less energy. Energy was lost. All right, so this is the amount of activation energy needed for the chemical reaction to occur without an enzyme. So if there's no enzyme present, this is how much energy would be needed to start this specific chemical reaction. Now, with an enzyme, you start with the same amount of energy with the reactants. You put in energy, but now with the enzyme, it makes it that you only need this much energy for the reaction to occur. So you can go like that. Okay? So if you actually look at the amount of energy needed from here to here, that's the activation energy with an enzyme. So you can see you need a lot less energy for this chemical reaction to start and progress versus without an enzyme. All right, so 3 important things about an enzyme. The biggest, most important thing about an enzyme is the enzyme's shape. The enzyme's shape is key for it to be able to do a certain chemical reaction. So if you remember, DNA has instructions on how to make your proteins. Enzymes are a type of protein. So DNA has instructions for how to make your proteins. DNA gets rewritten into mRNA during transcription. Then that mRNA is read during translation, and you make a protein. So all the way back here DNA has instructions on how to make the protein, and those instructions say what amino acids to connect together. So remember, amino acids, you connect together a whole bunch of amino acids and you're building a protein. And there's, remember, 20 different amino acids altogether. And the order that you put them in and the number that you put together builds the protein. And it's very, very specific. So a protein will be a certain shape based on which amino acids you put together in which order. So it's very, very, very important. This is why mutations can be bad, because mutations could spell out instructions that put together the wrong amino acids, and as a result of that, the enzyme doesn't come out the right shape. So the shape of the enzyme is the most important thing about the enzyme. So that's its 3D structure right here. So the shape of the enzyme, its tertiary structure. And that's determined by the DNA instructions, which say exactly which amino acids to put together in which order. When you look at its overall structure, you will notice the active site. This is where the substrate actually binds. So this is where the reactants that are involved in whatever chemical reaction this enzyme is going to be involved in, that's where they will bind. And some enzymes even have allosteric sites as well. Okay, and I'll talk about that in just a moment. All right. So first, the enzymes and their substrates. Just like the shape of the protein of the enzyme is very important, the enzyme and the substrate, the way they fit together, is very specific. So remember, that was one of the most important things, the two key ideas. Enzymes are very specific, and they help lower activation energy. So here, enzymes will only fit with certain substrates. So the enzyme you use in one chemical reaction can't be used in another chemical reaction. So there's the lock and key model that we see here. So we could see enzymes. So here's this enzyme. It'll only work with these substrates. You can't take this substrate and use it here, or this one and use it here. This enzyme will only use this substrate, won't use the other ones, and so on and so on. So we say enzymes are very specific and will only bind certain substrates. This lock and key model is really good for understanding specificity. Okay, so it's really good for helping us understand specificity and the fact that enzymes will only bind with certain substrates, but that's not quite how enzymes work. So that's really important right there. Okay, but it does help us understand that enzymes are very specific. This is how enzymes actually do work. So it's called induced fit. Induced fit basically tells us that the enzyme and the substrate really aren't a perfect fit. They're an okay fit. Okay, so here's a substrate, here's the enzyme, here's its active site here, and you can see that this substrate is not going to perfectly bind with this active site here. You can see the shapes don't exactly match, but they're close, and you can see the little curves here and there. So that closeness, what happens with induced fit, as the substrate comes to bind in the active site, the molecules and the atoms that are on this part of the substrate and the R groups from the amino acids that are part of the protein here, the enzyme here, are going to interact. And when they interact, they cause the shape of the substrate to change shape and the shape of the enzyme to change shape as well. So they change to fit each other even better. So it's an okay fit, but they change to fit each other even better. So that's induced fit. So that's what we see happening here. So lock and key model helps us with specificity. Induced fit tells us what is actually going on between a substrate and its enzyme. So they come and they bind together, and you have induced fit. And then we have the, here, enzyme substrate complex. So when an enzyme binds to its substrate, it's going to help lower activation energy in several different ways. We'll see in just a moment. And here's another way of looking at it. So here, this one, you'd probably be breaking this apart and you'd leave with like 2 or more pieces. Here, so that would be a catabolic reaction. Here's an anabolic. You take 2 and you're putting them together here. So when you see this, you can see the shapes aren't exactly a match. So induced fit occurs, so that way they fit together. Substrate enzyme complex, and then you have the release of the product. Notice, key thing here, the substrate is changed. The enzyme is not. So you can use the enzyme over and over and over and over again, because once it's formed the product, it releases it and it goes back to its original starting shape, and then more substrates can come, and that same enzyme can be used multiple times over and over and over again.